This figure is showing a human karyotype, which is basically an organized picture of the chromosomes that are in the cell. Now let's talk a little bit more detail about what we're seeing here. We said in humans, a normal non-reproductive cell has 46 chromosomes. And so if we look at our count, we can see we've got the numbers 1 to 22 with 2 chromosomes within each number. So that means there's a total of 44 chromosomes shown there. Numbers 1 through 22 are referred to as the autosomes, meaning they're non-sex chromosomes. The 23rd pair of chromosomes is referred to as the sex chromosomes. And in this particular case, we're seeing an X chromosome and a Y chromosome. So the sex of this particular individual would be male as determined by the sex chromosome content. If we had two X's, then the sex of the individual would be female. Now, within each number, we have 2 chromosomes. One of those comes from mom and the other one comes from dad. And so these 2 chromosomes within a pair are referred to as homologous chromosomes, meaning these chromosomes are similar in size, in centromere location, and the genetic content contained. So for example, on one chromosome, if there is an allele that determines blood type, it's going to be on the chromosome from mom, and it's also going to be on the same chromosome from dad. Now there may be different forms of the alleles present, but the similar genetic information is going to be there. So the karyotype shows us the arrangement of the chromosomes starting from largest to smallest, with again the sex chromosome content being last. In females, there are said to be 23 pair of homologous chromosomes because of the 2 X's. In males, technically speaking, there are 22 pair of homologous chromosomes and the XY. There is a small region of homology or similarity between the X and the Y chromosomes in a region called the pseudo-autosomal region. Now on a test, I won't try to confuse you with this. We'll just say in humans, there are 23 pair of homologous chromosomes, but I just wanted you to be familiar with that possible distinction for males. The colors that you see on the chromosomes are produced by staining, and these staining patterns help to identify homologous chromosomes, because when the chromosome preparation is made, the chromosomes just don't pop out in this nice organized array. You get kind of a smear of chromosomes. The individual doing the karyotype analysis takes the picture, and then they have to digitally move the chromosomes to where they identify the homologs. So you can see that chromosomes 1 and chromosomes 2 are fairly similar in size, but their banding patterns produced are very different. So these banding patterns, again produced by staining, help to identify the homologous pairs. Now, there can be abnormalities at the level of the karyotype, and one of the most common that people have heard is Down syndrome, which is also called trisomy 21. If an individual had Down syndrome, then their karyotype would frequently show 3 chromosomes for number 21. Again, that representing an abnormality. And the physical consequences of that abnormality are very evident in an individual with Down syndrome. So again, a number of aspects about karyotypes and the information contained within the karyotype.